Okay, let's talk about transcription. Transcription is a process of transcription is a process of copying DNA into RNA. That is transcription. It occurs in the nucleus and is mainly done by an enzyme called RNA polymerase. Okay, so let's show you. Let's try to sketch how this works. So, if we have a gene, so remember your DNA contains genes. All right. So, say here we have a double-stranded DNA. I'll just make the strands wide apart. Okay, double-stranded DNA, and if this strand up here, say, has this sequence of nucleotides, TA, G, C, A, T. All right. And then, of course, the complement strand will have A, T, C, G, T, and A. So it's a double-stranded DNA with complementary base pairing. Now, on this DNA, we're gonna make this part here. We'll call it this part the gene of interest to us. So this is our gene here. That's the gene that we want to copy into RNA. So in the DNA, upstream from the gene, you'll have a sequence of nucleotides that we call the promoter. And after the gene, you will have a sequence of nucleotides that we call the Terminator sequence. So each gene in your DNA is surrounded by one side has a promoter, one side has a terminator. And that's how you divide genes, one gene from another gene. They have these things around them that ensures that you can say copy this one gene without copying 10,000 other genes at the same time. Okay, so each gene will be capped by a promoter and a terminator at one end. So when the cell is about to transcribe this gene, the DNA, into RNA, the first step is. Step one is, so steps of transcription. Step one, RNA polymerase binds to the promoter. Okay, so the cell will reveal the DNA part of interest, and then the RNA polymerase will seek out its promoter. All right, and so once it's exposed, then the RNA polymerase will bind here to its promoter, and the promoter region can be something like a sequence of nucleotides. It can be ta or ta, we call it boxes, or very, very common sequences used to be for promoters. And so this is your RNA polymerases here, okay? And its job is to bind to the promoter and then copy the DNA into RNA. So here's step one. RNA polymerase binds to promoter region, and then in step two, it will copy a complement to the nucleotides on this strand, what we call the sense strand. The sense strand is a strand that has the promoter, by the way, okay? And this one will be called the antisense strand. So here again, if we did this, redraw, and we'll only draw the top one. So, but the bottom one is still there, of course. So you have the ta-ta, G, G, C, C, A, T, 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 T. So the RNA polymerase will move along, okay. And as it's moving along, right behind, as it moves along, it will copy the DNA into RNA. So here you get a C, a cytosine RNA nucleotide, a C, a G, and U. So that becomes the RNA that's copied from the gene part of the DNA. That's step 2. RNA polymerase copies DNA to RNA. Right? So wherever it sees a G, it puts a C. Wherever it sees a it puts a U. Okay? And vice versa. Now step 3 now, after it finishes, after it runs along, of course, these can be thousands of nucleotides long. So once it's done with the gene part, it will encounter a sequence of nucleotides that we call the Terminator. Okay? So then RNA polymerase reaches the Terminator portion of the DNA. So step 3 here. Again, so now it's down here. Here's step 3. It's here amongst the Terminator part of the gene, of the DNA. Of course, and you still have your RNA strand being built along the way, okay? And once it gets here, in step 3, it stops transcribing. And the RNA species is then released to be further modified. So at step 3, RNA polymerase reaches terminated portion and stops. At which point it stops transcribing. Okay. So now this thing that's formed is now called your pre-mRNA. Okay. So let's look then at how the cell deals with the pre-mRNA. So the pre-mRNA that's just made by the RNA polymerase, that pre-mRNA will contain regions that's coding and regions which are non-coding, meaning it has some junk in it, basically. Okay, so here, and maybe here. These parts, we call them introns. Those are sequences of nucleotides that will not end up in the final product. They're just there as fillers or as part of the extra DNA that's not used to make proteins. And then these parts, the other parts of it, are the exons. And the exons are the ones that will code for the real gene. So let's call them exon 1, exon 2, exon 3, exon 4, okay? So once you make the pre-mRNA, then the cell uses an enzyme to remove the introns. So here, to processing pre-mRNA, step 1 is enzyme removes introns, okay? And then step 2, enzyme joins the exons together. In various ways. For example, it may, once it pulls out the introns, it may say join E1 to E2 to E3 to E4. So it may do this 1, 2, 3, 4. That's how it joins segments. Or it may do 1, 3, 2, 4. Or it may do 3, 2, 4, 1. There are many ways that the cell can assemble the exons together. So this is called alternative splicing. And depending on how it combines them, each end product will code for a different protein. So here you see where you have one gene can give you different proteins because based on how it's put together after the pre-mRNA is modified. Now, once you have just exons in the pre-mRNA, these are called, so once you have just exons left over, then these are called mRNA. And this is what will be eventually converted into proteins. All right, now some housekeeping information here. So again, transcription, some facts about it. Occurs only in the nucleus, or predominantly in the nucleus. Again, done by RNA polymerase. And you may have several RNA polymerases may follow each other on the gene to make several copies of the mRNA. Several copies may work simultaneously behind each other. So that way you're making thousands of copies of the RNA, which will then give you thousands of copies of the protein eventually. So several RPs work on the mRNA simultaneously. 3, 1, 2, 3. Another fact or feature about this process is that your RNA polymerase will unwind DNA as it goes along. So it unwinds or unzips the DNA around 17 base pairs at a time. So go like this, unzip it, copy it, refold it, move down, unzip, copy, refold, and move down the chain until it hits the Terminator portion, okay? So unzips DNA around 17 base pairs, and it also rewinds the DNA afterward. After it has moved on, it will rewind behind it as it moves along. Okay, so that is a process of transcription. Okay, again, so know from me how it takes place and what enzyme does it, and how the pre-mRNA is different from the mRNA, and know of course what an intron is, what an exon is as well. Okay. And then once you then make the mRNA, then this is what is shipped out into the cytoplasm to be translated. So this is then translated, sent to be translated by the ribosome. Okay, all right, we'll pause there.